So just quickly, if we take a look at a comparison of these advanced models here, spheroids, organoids, and organ chips. So spheroids, these are typical monoculture 3D cell aggregates. They're great for screening for anti-cancer drugs, the efficacy there, as well as toxicity. They can be generated rather consistently, so you can get to medium to high throughput with these, but they don't have any mechanical forces or microphysiological forces. So in terms of contrast with the other two advanced models here, they're relatively low on the biological complexity and relevance scale. Organoids are a step up. You have a more heterogeneous population of cells. You're able to more adequately model disease. You're able to utilize these for personalized medicine and efficacy testing. The throughput isn't as high as spheroids, as they're a little bit more variable in terms of how you generate them. And they also don't have mechanical forces. Organ chips, these are microfluidic devices that recapitulate that tissue vascular interface, and that is a key difference here compared to spheroids and organoids. That is what organ chips are actually modeling, that vascular tissue interface. You can use any cell type in an organ chip, and you're able to capture mechanisms that you weren't previously able to capture in these simpler systems, which means you could do complex disease modeling and high fidelity toxicity testing. And again, with that flow and stretch of those mechanical forces, you're able to push that biological complexity forward and that in vivo relevance forward. It's not an either-or proposition here. Each of these in vitro platforms has utility across the drug discovery paradigm here. And really, it comes down to two questions. What is the biological question you want to answer? So what relevance or complexity do you need? And what throughput are you looking to get out of the system as well? So each of these systems, be it 2D cell culture, spheroids, organoids or organ chips, has their sweet spots in how they're being utilized in the drug discovery process. 2D cell culture and spheroid, because you can scale these up in a high throughput scenario, good for answering simple questions. They're more suited for screening and hit to lead. Organoids, because you're able to push that relevance forward, you're able to capture more mechanisms, tend to be used in the target ID validation, lead optimization, preclinical safety space. And again, a further step forward with organ chips, you can push that relevance even further forward and capture mechanisms that you just can't capture in organoids or any of these other systems. Again, really good for target ID validation. At the tail end of lead optimization when you're looking to pressure test your vital few compounds to move forward, and in preclinical safety. And of course, all of these model systems can be used in basic research as well.